Welcome to this latest podcast from Exeter Expertise. Today we are looking at the work adapting the mosquito gene in an effort to combat the spread of malaria. We'll be considering the wider implications of scientific breakthroughs such as this, and how should they be managed, including the governance, ethics, and how different stakeholders are engaged in the process. We're joined by three of the team involved in this work, and I'm delighted to welcome them onto Exeter Expertise today. First of all, we have Professor Sarah Hartley, who is an interdisciplinary social scientist at the University of Exeter Business School. She's joined by colleagues from Uganda, Stella Neema, an associate professor of medical anthropology at the Department of Sociology and Anthropology, School of Social Sciences, Malaria University, and her colleague, Chris Opesen. He's a lecturer at Malaria University and a postdoctoral fellow at the University of Exeter. All three of you, very warm welcome. Thank you for joining us today. Perhaps I could start with an obvious first question. Please tell us about the scientific efforts that are being made to alter the gene of the mosquito, and why is this work being done?
Thank you, Toby. This work is being run by Target Malaria, a group of scientists across the globe led by Imperial College in London. And this group of scientists with expertise in the Uganda Virus Research Institute developing what we call gene drive mosquitoes. These are like genetically modified mosquitoes, but they have a gene drive attached to them which forces the genetic change through subsequent generations. So essentially it drives the genetic trait. And at the moment, those traits are either to make the mosquito sterile or to stop it from transferring, being a vector for malaria. And if it is to make it sterile, it does that by biasing the offspring so that only male mosquitoes are born. So the purpose is essentially to eradicate malaria. It's the Anopheles gambiae mosquito on which they're focusing, and they're looking at 4 countries in Africa, and that's Uganda, Mali, Burkina Faso, and Ghana. Now, we're a group of social scientists. We're not attached to Target Malaria. We collaborate with them loosely. We're funded independently by the British Academy and by the Wellcome Trust. Understand the challenges, the social science challenges around this kind of technology development. So we're interested in governance. And by that, we're talking about things like risk assessment, democratic decision-making around whether we should or should not use this technology, how local stakeholders and communities can be involved, what consent means for a trial. These are the sort of critical social science questions which need to be investigated. But also, it's really important that they're investigated by independent social scientists, and even more important, by social scientists who are in the countries in which these trials may take place. And that's where our collaboration has been so powerful between the UK and Uganda, where this technology is being developed.
I guess everyone would agree very much that it's really important to try and stop malaria. It's a very worthwhile cause, but I guess people are less knowledgeable about the unintended consequences or the different groups that need to be consulted around that. I mean, it sounds like a very complex set of issues that you were dealing with there. So could you just talk us through some of the ethical and governance challenges?
One of the big challenges is the fact that this is the most cutting edge biotechnology at the moment. And it's going to be developed and rolled out in some of the countries, particularly countries like Mali, where there is extreme poverty, extreme disease, and where there certainly is a historical legacy of considerable imbalance of power in relationships between researchers and those in those countries. So we're dealing with strong neo-colonial histories. What we're seeking to do is to open up decision-making around these technologies so that people living in the countries experiencing malaria are able to feed in and shape the direction of this technology. That's really where we're looking to expand the range of voices and values that shape technology.
Thanks, Sarah. And I guess the sort of obvious question is to explore a bit more deeply, why do you need to do that? So if you didn't do this work and the actual pure science went ahead, what are some of the risks attached to this project?
There are 2 main drivers for this approach to opening up governance of science and technology. One is democratic and the other is what we call substantive. So democratic reasons are that there is a democratic right for people to be involved in decision-making around technologies which have radical implications. Within our project, we don't see the divide between expert and lay or between scientist and public in quite the same way. We see people as having various different types of knowledge that can contribute to the development of this technology. And so for us, there's also a substantive reason for engagement and for inclusivity. And that is, we believe that you can make a technology better by bringing in the knowledge and experience of those people who are experiencing the problem the technology is trying to solve.
So this is very much about what people would understand by stakeholder management. Chris, can you give us an insight into the different stakeholders that you've been talking to and the work that you've been doing with them?
So far in our collaboration, we have done 3 studies on gene drive in Uganda. The first one, and the first one of its kind in Uganda and probably entire sub-Saharan Africa, where all these trials are ongoing, was themed co-developing risk assessments across disciplines and borders, focused on gene drive in Uganda. So in this study, we had an engagement and interviews with an array of stakeholders, and this included the specialized scientists that are involved in research at Target Malaria, that is at the Uganda Virus Research Institute. We also talked to regulators, academia, representatives of the civil society, environmentalists, local government representatives, and community representatives. During these interviews and engagements, we found that most of the current focus in research is heavily on concerns and not hopes. As our point of departure, we note that there is need to focus on both hopes and concerns. Now, in these engagements that we've had with these different stakeholders, the hopes have been raised around 2 areas. One is on the ability of gene-drive mosquitoes to address malaria. And the hopes that are raised under this area are that, one, gene drive might actually be the effective solution we've been looking out for to reduce or even eliminate malaria. But then the other second hope that stakeholders have is that gene drive might become another tool in our arsenal in the fight against malaria. Now, on the second area, that is the socioeconomic benefits associated with gene drive, the hopes that stakeholders raised that gene drive might help us to increase productivity. That is, if it helps us to eliminate or reduce the burden of malaria. And secondly, that from gene drive, we might be able to save a lot of money that we're spending on treatment for malaria. On the other hand, ecological impacts of gene drive testing and deployment were raised as the main concerns with the gene drive. And in particular, concerns raised were that, one, gene drive caused a decrease in non-target organisms in the ecosystem, given, of course, the food chains. The fish might be eating some mosquitoes, the birds might be eating also, and so on and so on. Secondly, that gene drive might also impact on the non-target organisms through vertical and horizontal gene transfers. Now, the paper that we have published on hopes and concerns talks a lot about these issues, and we can easily access it on the Malaria Journal. We recommend that for more details on these concerns and hopes, we can look out for this paper in the journal.
A fascinating insight there. And it says to me, I think, that obviously it's a very noble cause to try and alter this gene, but as in a lot of science, actually, there's a whole host of potential unintended consequences. And I guess, to what extent in your work are those unintended consequences managed? Do you feed this information back to the scientific community and the other stakeholders? How does the sort of practical process work?
When we spoke to the different stakeholders, you could see a lot of pure science and goodwill. Everybody was interested in producing a risk-free product. And to ensure that this happens, all the protocols that are involved undergo, for example, a critical process of risk review. And this starts with an institutional review board, the IRBs, institutional review bureaus that are based in the research institutions. So the researcher develops its proposal, subjects it to review by this board. And if this board passes it, then it's taken to the National Environmental Management Authority. And then once the National Environment Management Authority, in short, Neema, reviews it, it will categorize it in terms of risk, either in category A, category B, or category C. Once this categorization is done, then a fully detailed environmental impact assessment is conducted. Now, a report from this assessment, together with the report from the Institutional Review Bureau, then sent to the Uganda National Council of Science and Technology, where again a specialized committee known as the National Biosafety Committee will review these two reports. Once they are approved, then the project is sanctioned to go ahead, but usually with the terms and conditions that the project team must observe. And then also the members of this committee have to follow up the implementation team to ensure that they comply with the terms and conditions that accompany the approval.
I'd like to bring Stella in at this point because, Stella, it'd be interesting to ask you, based on this project, what are some of the lessons that you've been able to draw out for future social science projects, whether that's on gene drive specifically or indeed at other scientific areas?
Several lessons were really learned. This was something new to the participants. First of all, we learned after they did their group discussions and listening to what the keynote speakers were talking about, we learned that inclusiveness is very important. It was seen more, especially where we need to include researchers, the local researchers, not only the researchers from the north. So there was we needed cross-fertilization, but also with communities, with CBOs, with NGOs, so that we include as many people at stake as possible for especially even the end users for acceptability. And there were also fears of safety. So what did this mean? We also learned that we needed a lot of preparedness. Preparedness is key on the part of the scientists, both the local and also the external, but also to the government, including even the regulators. That preparedness seemed to be starting. Yes, we have the regulatory framework, guidelines, laws, but some of them are still bills. They haven't been enacted. So it's work in progress. And then finally, we realized that the general information is lacking about the gene drive. So we felt that there was a lot of skepticism. People were just fearing instead of thinking about a better solution. So we thought that maybe we needed a lot of sensitizing people about this so-called gene drive.
And I think building on that, a question, I guess, to all three of you, actually, based on the work that you've been doing in this area of the world, what do you see as some of the, I guess, likely future challenges, mainly in terms of the social science?
I think I'll take that one on. One of the challenges is that regulation will need to evolve with the technological advancements, stages in the gene drive development process, and risk mapping. The second challenge is that building public and political support that currently is hindering development and deployment of gene drive mosquitoes remains a huge task, but we still think that there's a way through. For example, we think that it's necessary to make engagements on gene drive more inclusive, bring in all the different actors, the different stakeholders, and then we should be able to win both the public and political support and eliminate the as a current obstacle to the deployment and all the trials that are associated with this process. And to contribute to this, we are working on another evidence-based publication paper on who should be involved in engagements on gene drive risk assessments. The stakeholder views on this assessment indicate the need for inclusion of a broad spectrum of stakeholders. And this includes project-affected persons and communities, regulators, scientists, the public in general, the civil society, academia, and the politicians using the different platforms.
I guess a key question there is, do you see some of the challenges that you've experienced in your work being unique to Uganda, or can you translate some of the lessons from the research into other areas of the world where actually, there's a lot of work going on in adapting genes? Some people will be concerned about. I mean, how translatable is this to other parts of the world?
I see that this technology is novel across the world. And so the perceptions and the interest for every stakeholder wanting his views to be represented in the decision-making process are cross-cutting. Given that what we observe here and what we have been seen in Mali and Burkina Faso is quite similar, I think that these challenges represent the realities beyond the boundaries of Uganda. And that means that they can actually be generalized.
I'm going to add to that. We have a number of projects that are looking more broadly at gene drive governance outside of Uganda as well. We are working with Target Malaria on a paper looking at engagement in risk assessment. Now, gene drive is an unusual scientific space because there is a huge commitment to responsibility by those developing the technology. They're acutely aware of the kind of ethical issues involved, and they have reached out on numerous occasions to collaborate and to understand how they can do engagement better. So the kind of work on the ground that Stella and Chris are doing in Uganda is just critical for Target Malaria to do a better job, but not just Target Malaria. There are other gene drive developers with interest in India, for example. These lessons may also be valuable for those developing genetically modified mosquitoes in Florida and in Brazil. So the cutting edge work we're doing around how to bring in these other voices and values, particularly in the areas of risk assessment and risk governance, is really transferable across to other technology areas as well. There really is a lack of social science input into these spaces around risk assessment and engagement.
Building on that, Sarah, why would you say this hasn't happened before? What have the barriers been?
One of the barriers has been that non-experts have seemed to view non-experts as having very limited contribution to technical questions. And I think that has changed over a number of decades. So thinking around from the 1980s in the UK, we had the Bodmer report, which was a report which looked into the relationship between science and society and talked about how the public was perhaps losing trust in science. And so there was a need to educate the public about the value of science. That shifted, I think, with conversations around biotechnology and genetically modified crops to view trust as really critically important. And then we needed to have a dialogue with publics. But still, it was seen to be publics having less knowledge and rationality than the experts. And I think that is starting to change with this idea of knowledge co-production and the idea that knowledge is co-produced and it's not just experts who have an authority on knowledge, particularly in this kind of project where field trials will likely take place in Uganda, Burkina Faso or Mali before they will take place anywhere else in the world. And that knowledge in Uganda from communities and from stakeholders can help to localize and contextualize the technology. And I think we're moving as a society towards that recognition of the value of different types of knowledge. But risk assessment is a highly technical space. It is the most technical space when we talk about governance of technology. And so it's been a hard nut to crack, but I think we're making progress.
I think that's a really good place to finish. It's been great hearing from all three of you about this very important work. So thank you very much for spending some time with us today.
Thank you.
Thank you very much, Toby.